All right, DNA structure and DNA replication. Hopefully remember from the other day, base pairing rules. When DNA is copying or replicating itself, the base pairing rules, all that means is, remember, adenine or A, do you remember what it bonds with? Please tell me you do. It bonds with thymine or T. Cytosine or C always bonds with guanine or G. Now, every now and then, DNA does make a mistake when it's making a copy of itself. So, maybe it bonds a C with a T or something. It makes some sort of mistake. And anytime it makes a mistake when it's copying itself, that's called a mutation. Now, most mutations in our DNA, you don't even notice it. They're called silent mutations, or they're neutral. They don't hurt you. Every now and then, if your DNA mutates so much, it can be lethal, and it can cause things like cancer. So we're going to see how DNA makes exact copies of itself. And remember, DNA holds the code for building proteins from the monomers, or the building blocks of proteins, amino acids. All right, continuing on, DNA replication. The first step, or one of the first steps, when DNA has a shape like a ladder that's been twisted around. Remember, on the outside of the ladder is where you put your hands. That'll be where the sugar phosphate on both sides. Sugar, phosphate. And remember, the inside where you'll put your feet, that's where the bases are, the A, T, C's, and G's. So the first thing you got to do, just like every day during the winter when you unzip your sweater, and you hold on to the little handle, and you unzip your sweater, well, that's the first thing we're going to do here also. That little part that you hold on to unzip whatever you're unzipping, that's an enzyme called helicase. And that guy is right there. Helicase. Helicase. And that guy goes right down the middle of the ladder, basically goes right down the middle of the DNA, and it unzips. It breaks the hydrogen bonds between the A's and the T's and the C's and the G's. So just like unzipping a sweatshirt or whatever, helicase, this enzyme, comes down and unzips, breaks the hydrogen bonds between the bases. Then another enzyme is going to come along, and it's going to add the other part of the zipper. So an enzyme called DNA polymerase number 3, so we can just call him polymerase 1, 2, 3. He comes, and all he does is he grabs free-floating nucleotides and attaches them to the exposed bases. So with this A right here, he's going to grab a T and put a T there. With this T, he's going to grab an A. A goes with T. C goes with G. G goes with C, and so on. It goes down the line. DNA polymerase just puts a nucleotide with the appropriate base whenever it sees fit. So G goes with C. A goes with T, and so on. Last one, G goes with C. Now, DNA polymerase number 3 can only work in one direction, and that's called 5' to 3'. So if DNA polymerase is going this way on this strand, polymerase has to go the opposite way on what's called the lagging strand. This will be the strand down here. So it's going to have to go in reverse. It's going to put an A with the T, a T with that A with that T. So it did a little chunk right here, a little chunk of DNA. It allows helicase to keep unzipping, and then it keeps adding nucleotides in little chunks. So with this A, it's going to put a T. With this C, it's going to put a G. With this G, it's going to put a C. That's another little chunk of DNA on this side of the DNA double helix. With this C, it's going to put a G. With this G, it's going to put a C. And with this A, it's going to put a T. So that's another little chunk. So the difference is, on the top strand, DNA polymerase can go continuously. It never stops because it's replicating DNA the same way that helicase is unzipping the DNA. But on the bottom strand, what's called the lagging strand, it has to put DNA in little chunks, and these chunks are called Okazaki fragments. That'll be extra credit for regular bios honors. Sorry, you're going to have to know that. We'll go over it more here in a second. All right, DNA replication, the process by which DNA makes an exact copy. All right, some real quick replication facts. It is super complex, but we're going to try to make it fairly simple. It happens pretty quick. Your DNA can't copy itself in about an hour. And it's, copying a DNA molecule is not like when you guys copy homework assignments off each other, like 1 or 2 pages long. These things are 200, 250 million base pairs long, so it's pretty long molecules. It's very accurate. It very rarely makes mistakes. And remember, when it makes a mistake, that's called a mutation. You've probably heard of the Teenage Mutant Ninja Turtles or mutants. Well, mutation, that's when your DNA gets altered. It requires a bunch of enzymes. Remember, enzymes are special proteins that speed up chemical reactions in cells or in the body. So we're going to learn a handful of different enzymes that helps DNA replicate or help DNA make a copy of itself. All right, so why would a cell need to replicate its DNA? This is ridiculously simple. Whenever cells reproduce, or whenever you're going to make a new cell from one cell, you got to have a copy of the DNA so both cells can have their own set of genetic material, their own DNA. So every time one cell is going to divide into 2, we need new DNA. DNA needs to replicate. Remember, that happens in the S phase, or the synthesis stage, of interphase. All right, so step 1, DNA, which is kind of like a twisted staircase, twisted ladder. First, it needs to uncoil and become kind of like a straight ladder like this. Now remember, once again, on the sides here, this is the sugar and the phosphate, the sugar and the phosphate. Same over here too. The deoxyribose sugar, phosphate, sugar, phosphate. And here inside here, these are the bases. So these are the bases like the A, T, C, and G. That's here in the middle. Remember, it'll be the second step. First, the staircase untwists and becomes a straight ladder. And remember, the enzyme helicase is going to come right through the middle, and just like you unzip your sweater, helicase is going to go right through the middle, break those hydrogen bonds, and create 2 halves of the zipper. So it'll be one over here, and it'll be one over here like that. So helicase comes through and unzips that DNA molecule. All right, here's another picture that shows about what I just said on the last slide. So remember, the enzyme helicase. That's the guy that's coming right down the middle and is unzipping. So this A right here used to be bound to this T right there. This C over here used to be bonded to that G, but they've been unzipped by helicase. Went right down the middle and broke those hydrogen bonds. Now remember, polymerase 3 is going to come down and attach nucleotides where they go. So with this G, it'll put a C. With this A, it'll put a T. And on this side, the leading strand, polymerase can just follow helicase all the way down. But remember, on this side, the side over here, the lagging strand, polymerase has to work in reverse. So it has to go this way, it has to go down. So it puts an A with that T, a G with that C, a T with that A. And it does it in little chunks of DNA called Okazaki fragments. Regular bio, a lot of extra credit for you if you want to earn it. Honors bio, some extra credit for you too, but not quite as much. All right, the exact same picture as last time. So now that helicase, and I'll draw him in here again, not that he looks like that, but he or she, it's not really a he. Helicase unzips it, and it leaves what's called a replication fork, or a template. So it leaves these exposed bases. And polymerase 3 just comes along, and like I said in the last one, just attaches free-floating nucleotides, like these guys that are floating around out here, Those are nucleotides. Remember, that's a phosphate bound to a deoxyribose sugar and bound to one of the bases. So this is the deoxyribose sugar, and that's the phosphate. So that's what these guys are floating around out here in the nucleus. So polymerase 3 just bonds these bases where they go. C with the G, T with the A, C with the G, etc. And it just follows all along the way. And that guy, once again, is called polymerase 3. All right, so where are these free nucleotides found? Well, of course, they got to be found in the nucleus, because the nucleus, that's where our DNA is. So if our DNA needs to make a copy of itself, it's going to need a lot of DNA parts. And remember, those parts are nucleotides. So we need those parts to be in the nucleus so we can make new DNA. What does the word complementary mean? That's easy. That just means this. A goes with T, and G always goes with C. T always goes with A, C always goes with G. Or, and here it is again, adenine always pairs with thymine, or T, and cytosine always pairs with guanine, or G. The base pairing rules, always put B, P, rules. Ensure that mistakes called, once again, those are called mutations. Mutations are somewhat rare. Alright, so this little picture can be a little bit misleading. It's not totally accurate. So up here, this is where the DNA has not been unzipped yet. You can see that T and the A and the A and the T, they're still bonded together. So helicase would be like right about there, and it's coming down and it's unzipping it. And you can see here where polymerase number 3, the enzyme, is already starting to add bases to these exposed bases here. So it would put a C, is going to put a T there. Now remember, on one strand, it can go, called the leading strand, it can go the same direction as helicase. But on the opposite side, it's got to go the opposite direction as helicase moves. So in that one, it's got to put it together in chunks called Okazaki fragments. But what I want to show you here is, as you can see, this strand of DNA out here and this strand out here, the two on the very outside, those are the original strands. So A out here and D out here, A and D, that was our original strand of DNA. We didn't make that. All we did was we unzipped it when helicase came through here. So A and D, that's old. That's old DNA from our old DNA molecule. Now, in here in the middle, B and C, those are brand new bases being added to the DNA molecule. So this, the inner sides, the B and C, that's brand new DNA being made by DNA polymerase 3 as it adds bases to its complementary base. T with A, C with G, G with C, and so on. Done it. All right. Earlier I said that DNA has what's called a 5 prime. They just put a little dash there for 5 prime end. And it's got what's called a 3 prime end. And that's really pretty simple. As we can see here, the little red ball right there, the little red ball right there, that's a phosphate. If the DNA ends with a phosphate, the little red ball, that's called the 5 prime end. This, as you can see on the other side down here, it ends with a deoxyribose sugar, that little blue little house-looking thing, that's a deoxyribose sugar. If it ends with a sugar, that's the 3 prime end. So this one, you can see it ends with a phosphate, so that's going to be the 5 end. And up here, it ends with a deoxyribose sugar, so that's going to be the 3 prime end. Now, why do we care about that? Well, DNA polymerase 3, the enzyme that's going to add the bases, so the C, it's going to put a G there. That guy can only work from 5 prime to 3 prime. So, on this segment of DNA, polymerase 3 can move up this way and down this way, because as he adds this G, he's also going to add a sugar, and this one will have a phosphate sticking off. This side does not, this one will, just like over here. It's always the opposite. If this side does not have a phosphate at the end, this side will. If this side up here has a phosphate, this side up here will not. So polymerase 3 is going to follow, because remember helicase, the enzyme that did the unzipping, it's moving this way. So polymerase 3 is going to come, and with this C, it's going to add a G. And it's also going to put another sugar, another phosphate. With this T, it's going to put an A. With a G, it's going to put a C. With the A, it's going to put a T. And with the C, it's going to put a G. Now, on the other side, polymerase cannot go the same direction as helicase, because it has to go from 5 to 3. So on this one, it's going to have to go down. So with this A, it's going to put a T. With a G, it'll put a C. And with this G, it's going to put another C. So it's working this way. And it's going to put these segments of DNA together on what's called the lagging strand. This is the lagging strand over here, in little chunks called Okazaki fragments. So as helicase continues to unzip the DNA, polymerase 3 will keep coming behind it and adding nucleotides after the bases are unzipped. So this is called the leading strand, because polymerase 3 can just continuously follow helicase, adding the bases. But on the lagging strand, it has to put them together in chunks called Okazaki fragments. All right, so a little diagram that shows some of the enzymes and what they do in DNA replication. So here's the DNA molecule that has not been unzipped yet by helicase. Helicase is moving that way, unzipping it. This out here on the very outsides, remember that is the sugar phosphate backbone. These are the bases in here in the middle, the A, T, C's, and G's. Now if you remember, DNA polymerase number 3, this guy right here, can only make DNA in the 5' to 3' direction. So you can see this is 3' up here. So this will be the 5' that's being constructed right there. So it's making it from 5 to 3. So on this side, this is called the leading strand. I'll put it in yellow. The leading strand, polymerase 3 can just work continuously, following helicase and attaching free-floating nucleotides to the exposed bases. So basically putting A's with T's, C's with G's, G's with C's, and T's with A's. That's what polymerase 3 does. Now, on this strand over here, since polymerase 3 can only go in the 5 to 3 direction, on the bottom strand, the lagging strand, it's got to go the opposite way, because 5 to 3 works in the opposite way on the lagging strand. It's the opposite way that helicase. Helicase is going that way, but the 5-3 on the lagging strand is going that way. So it can only lay down, polymerase 3 can only lay down DNA in short little chunks called Okazaki fragments. And there it is, Okazaki fragments. So you can see 3 of them right there. Now, another thing, RNA primase, that's another enzyme. It's got a pretty simple job. It lays down what's called a primer. Because polymerase number 3, the one that attaches the bases, it cannot hook onto the DNA unless there's a primer attached to it first. So primase attaches the primer, and then polymerase 3 can go down and attach the bases. So you can see another primer over here. Polymerase 3 came in and added these bases. Polymerase 3 came in and added these bases. There's another primer right there. DNA polymerase 1, its job is to come in and remove the red primer of RNA and replace it with DNA. So DNA polymerase 1 gets rid of these red RNA primers and replaces it with DNA. Now, the last enzyme we need to be concerned about is this guy right here, DNA ligase. As DNA polymerase 1 came in and got rid of the primer, you can see that it forgot to bond together the sugar and the phosphates. It forgot to do those phosphodiester bonds. So ligase comes in and bonds those nucleotides together so they're nice and strong. All right, so just here's the enzymes we're going to need to know. It's only 5. And for regular bios, these will be extra credit, and extra credit. Honors, sorry, you're going to have to know all of them, but they're not that hard. Remember, helicase, that's the guy that's going to unzip the DNA. DNA polymerase 3, that's the guy that's going to add the other part of the zipper. So once you unzip it, polymerase 3 is going to add the other half of the zipper on each side. Remember, polymerase 3 cannot start working unless RNA primase first lays down a primer or a starting block to give polymerase 3 a place to begin and attach. Remember, DNA polymerase number 1 is the guy that's going to get rid of the RNA primers and replace it with DNA nucleotides. And the last and final one is DNA ligase. Those are the guys that bond together the Okazaki fragments to create a continuous strand of deoxyribonucleic acid, otherwise called DNA. All right, when it's all over, you get 2 strands of DNA, which are exactly the same as the original strand. Now, a term you need to know, semi-conservative. I don't know if you can remember back about 7 or 8 slides, but remember when we start out with DNA, we start out with a ladder. I'm going to make this one red. That's DNA. Remember, helicase is going to come through and unzip that ladder. It's going to come through and unzip. So our ladder is going to start looking like this. It's been unzipped. Now, polymerase 3 is going to come in and add bases with the complementary bases. So if there was this right here is a T, that would be an A. If that's a C, it'd put a G. If that's a T, it'd put another A. If that's an A, it'll put a T, and so forth. So over here, it'd be a T, and a G, and a T, and an A. But, as you can see, as we make DNA, the red part of the DNA on the outside, so this red out here, that was from our original strand. So the outside, this is old DNA. And on the inside, that's brand new DNA that we just made. That's new. So that's all semi-conservative means. Half of the new strands are old from the original, and half of the strands are brand new, we just made them.